So that cellular energetics of the kind that we're looking at here becomes very important in thinking about cancer biology. I mean, it's only one aspect of cancer biology, but it is an important one. So, yeah.
So these three, like C, epsilon, and R,
Yeah.
should we just basically be able to know what, like if you're affecting an enzyme or changing an enzyme concentration, that's going to be changing the C, if you're
Right.
changing that, and so on?
Yeah. Yeah, I mean, certainly no more than the basic definitions and interpretation of what this plot means. Yeah.
So you said it's unexpected that hexokinase would be the main enzyme like control, right?
Yes.
It was unexpected, yeah.
So is there an explanation for why?
Not that I'm aware of. I mean, the thought was, you know, PFK is the kind of the committed step. So that would be naively where you would expect the regulation to be. What does it mean that the regulation seems to be more dependent on the earlier step? I don't have a quick answer.
Hexokinase is still an irreversible step, right?
Yeah,
So
yeah, it is.
that kind of makes sense.
Yeah, but you're comparing two irreversible steps, and the product of hexokinase can still go off into another direction. And yet, what you're measuring the output of this experiment is the flux through glycolysis. Right, so you're kind of looking at, well, how much pyruvate's coming out the other end, for example. So it's saying that turning on the earlier enzyme, you know, isn't really shunting off the stuff to the sides so much. You might argue that this is in an extract and not in an intact cell, and that might make the difference. I think that would be a pretty good criticism of this experiment.
I'd also like to know what the levels were for the side paths.
For what?
If the side paths
Yeah.
Also increase at the same level.
Measure how much is going on, not just for glycolytic flux,
Yeah.
but for what's going on in the other, yeah.
Because it would make sense that all of them would get increased.
Yeah, so sometimes I mean, you're also limited by what you can easily detect.
Yeah.
And how much you can detect at the same time, but it's a good point, for sure. You'd want to be able to see everything. I mean, a lot of what's going on in metabolomics is computational, that physicists types are programming this metabolic net in as almost like an electricity circuit, where you have lots of different interconnections between the nodes, which are the substrate molecules, and doing input and output analysis on that. And you know, one big problem with that is that we don't know what the whole circuit looks like yet, because you know, even in very, very well studied cells like E. coli, something like half the genes, we still don't know what they do. Right? I mean, it seems like we know a lot, and we do, but it's also worth, you know, paying attention to the fact that for half the genes, we don't know what they're doing. So that certainly severely limits, because there could be some gene that we don't know about that comes along and makes a protein-protein interaction with PFK or hexokinase, and maybe that's controlling, you know, what's going on, and we don't know anything about it. We're surprised about PFK because that gene somewhere on the chromosome, while it's been sequenced, we don't know what the protein does. Other questions? Yeah.
Is it not possible to put markers on every single protein that's made? And track all
Well, yeah, we'll know they're there.
I mean,
We'll know they're there.
Can you track them in live, in vivo?
To a limited extent, yes. You certainly can hook a tag on the protein, track it in the cell in different compartments. It's possible to deliver the tag. Yeah. So, but you don't know which one to tag either.
But if you tag every single one with something different.
How long do you want your PhD to last? I mean, it gets into the question of, you know, how much is really feasible.
Right.
So it's challenging to do that. I mean, you can, on a 2D gel, you can break open E. coli. You can look at this extract, and you can run, you know, 2 dimensions of a gel and separate all the spots if you really do it at a high enough resolution. And then you can mass spec every spot, or once you've done it once, we know where they run, so you can identify them. And then you can change, you know, you can add an inhibitor, an effector, and see which ones are changed in terms of gene expression. You can look at the messenger RNA. But looking at a protein-protein interaction, that's more challenging. Because as soon as you break a cell, it may easily break up if it's a weak interaction. So you don't actually detect it anymore. You know. So, and then if you want to tag something specific, you don't know which one to use. You don't know what to tag. So it's, it could well be proteins that interact with glycolytic enzymes, and there's nothing in the book about it because we just don't know. There's a lot we don't know. I remember when I first started graduate school, and I looked at a problem that my advisor assigned me, and thought of something, and I was like, oh, but surely, if I could think of it, surely somebody will have done that. And then I went to him, and he said, no, nobody's done that. Nobody's done this. Nobody's done any of it. Well, what do you mean no one's done it? But it's true, we haven't done it. Yeah?
Do these results change with the different isozymes of hexokinase?
Does it change with different isozymes?
Yeah, do the results change? Different
I
isozymes of
don't know.
hexokinase?
I don't know. Could. Okay, let's go on.
Nadia has a question.
Oh, I'm sorry, was there another question?
Yeah.
I'm sorry.
This previous.
We
I'm
love
still
this.
thinking about this PFK1.
Yeah.
Maybe this increase is because it is using product from first reaction, and then it reaches plateau because hexokinase could produce only as much of product. So I mean, like.
To what extent does it matter that PFK is coming after hexokinase in the pathway?
I mean, like you
Yeah.
can dump as much as you want PFK if you don't have input from first reaction, you will have this plateau. So it could be regulatory, but it doesn't have reactant
Yeah, I think
to
we would
start
need to
with.
look at how the experiments were done exactly. Like, does the experiment start by adding a whole bunch of excess glucose, for example, so that there's the capacity to have a high throughput, and then any enzyme might modulate it?
But if you have limited amount of hexokinase, it can do more than it can.
Right. Yeah. So there's not so much of the substrate ever being generated.
Yeah.
So, yeah, I think we don't know enough from how we know about this experiment to address that question. It's a good question, though, for sure. I mean, maybe this is sort of artificially low because of the way the experiment was set up, and it just doesn't have enough substrate. And if that's the case, then it's not well-controlled experiment. Right? Because the conclusion that is being drawn is that this is the most important enzyme for regulation. But if the experimental setup was that there's a problem because you didn't give it enough substrate to really see that, then maybe the conclusion isn't so good. Okay. All right. So this is interesting. So this is telling me to spend more time on this next time. So okay, we talked about why we cannot run both at the same time because of this. This is called a futile cycle, by the way, for obvious reasons. So let's look at some of these different enzymes and ask what's actually happening with them. All right. So we've got hexokinase, and we've got different isozymes of hexokinase in different tissues. So hexokinase 4 in the liver has a low glucose affinity. So the glucose concentration is, the Km is so high, so here we're using Km and affinity as equal. Right? We know from a mechanistic analysis it's not precisely equal to affinity, but this is what happens when you start to think about things like biology and regulation. That distinction between Km and affinity tends to fly right out the window. Right? And there's kind of no way to get out of that because we're not at that level of detail. It's just that you don't want to extend that fly out the window stuff if you're actually thinking about the details on an enzyme. Okay, liver enzyme, high Km, 10 millimolar. Why? Because the liver is generous and wants to give it away. Right? Glucose concentration in the cytoplasm, 5 millimolar. So if the glucose concentration gets really high, it will start to work so that it doesn't let the glucose in the bloodstream get much above 5 millimolar. But otherwise, it only